Interphase. The cell is functioning and performing its cell duties. The DNA is not being used and cannot be read because it is condensed. The DNA is also being duplicated.
During prophase, crossing over occurs. This is the act of the switching of genes of homologous pairs, creating genetic diversity.
Metaphase 1 is when the homologous pairs line up on the metaphase plate. During metaphase 1, the process of independent assortment occurs. Independent assortment is when the homologous pairs orient themselves randomly on the metaphase plate. This creates a mixture of paternal and maternal genes for the daughter cells that will be produced later on. This contributes to genetic diversity. There are many random possible outcomes for this. In humans, there are 2 to the 23rd possible outcomes. Anaphase 1 is when the homologous pairs split and are pushed by the mitotic spindle, created by the centrioles, to opposite sides of the cell. Their final formation is random and therefore increases genetic diversity. Telophase 1. 2 nuclei are being condensed and reconstructed into 2 new cells. The spindles are broken down. During cytokinesis 1, 2 haploid cells are formed. The DNA condenses. During prophase 2, the nucleus dissolves. There is a shortening and thickening of chromatids, and the centrosomes move to opposite sides of the cell and arrange spindle fibers for the second meiotic division. During metaphase 2, the duplicated chromosomes line up on the metaphase plate because of the push from the mitotic spindles for the second time. All movement of the chromosomes is due to the mitotic spindles. During anaphase 2, the sister chromatids split. Telophase 2 is the final phase of meiosis. In it, the decondensation of chromosomes and the disassembly of the spindles occurs. Finally, the final product is 4 daughter cells. Fraternal twins arise because during oogenesis, 2 eggs can be released, and when fertilized, they can be fertilized by 2 different sperms, therefore 2 separate people, but they are growing at the same rate and time. Because of the genetic diversity, the 2 eggs and 2 sperm have different DNA, resulting in fraternal twins.